All right. Why don't we get started now? So welcome everybody. My name is Rahim Kanji. I'm the Executive Director of the Ontario Water Consortium. Really appreciate everybody joining us today. The Ontario Water Consortium is Ontario's only sector support organization that's dedicated solely to advancing research and technology to address water challenges. We bring together researchers, companies, municipalities, governments, all sorts of stakeholders to drive collaborative R&D, pilot testing, and real-world technology adoption. We help bridge the gap between research and market-ready innovation. And while there are many different kinds of innovation organizations across Canada, none are focused specifically and exclusively on water like OWC is. Ontario Water Consortium's collaborative model has delivered measurable results. And over the past decade plus, we've helped mobilize over $90 million in project funding, supported or created more than 500 high-tech jobs to tackle water challenges, and advanced upwards of 80 water technology development and commercialization projects. We've partnered with more than 100 academic researchers plus students to help build infrastructure, to help advance technology, including building wastewater test beds in London and in Guelph. And we do all of this by connecting innovators with adopters, aligning the right funding, the right partnerships, all to help move solutions forward. Before getting into today's webinar, we also hope that you'll join us again for the second session of our Innovation Showcase Summer Series. So on June 9th, we will hear from researchers at the University of Waterloo and Western University on recent advances in the detection, characterization, and treatment of PFAS in Canadian water systems, including new analytical methods, non-target screening approaches, and integrated treatment strategies to improve monitoring and mitigation of forever chemicals in drinking water and wastewater systems. This session is free to join, and you can also note this QR code on the screen that you can use to subscribe to the OWC newsletter. And not on the screen here, but if PFAS is your thing and is your interest, then also know that Toronto Metropolitan University is hosting a PFAS symposium live in person on June 19th. Spots are still available. And for questions about this, fees, sponsorships, attendance generally, you can reach out to Angela Murphy. And I will drop the link to both of these, the webinar and the TMU event. I'll drop those links into the chat, as well as Angela's contact information. So I'll do that once we get started. But for now, I'm very pleased to introduce Dr. Christopher Lawson, who will begin today's webinar that's entitled Beyond Biogas. He just took my screen away from me here. That's okay. And we're going to look at, he's going to introduce us to an overview of the Waste to Chemicals Alliance and the integrated approaches being advanced to move anaerobic digestion beyond traditional biogas production and into the recovery of high-value chemicals from organic waste streams. Following Dr. Lawson, we'll hear from Jasmine Parmar, who will discuss the production and recovery of medium-chain fatty acids from source-separated organics. Ethan Agina will follow Jasmine, and he will present on advances in synthetic biology for expanding anaerobic fermentation. And then we'll hear from Dr. Jay Werber and his team, who will highlight membrane innovations for chemical recovery, followed by Diana Dushikanova, who will speak to inline extraction systems and their impact on medium-chain fatty acid production. And finally, Renzo Gutierrez will present on scaling up solvent-free membrane technologies for selective chemical extraction. Please hold on to your questions. We'll have time at the end for questions, so you can hold on to them, or if if it's something that you just don't want to forget about, just drop your question into the Q&A feature. Okay, so we're going to pass it on to Chris now, and we can see your screen. So I think you got a little bit of a head start, but it means you're ready to go. So on to you, Chris.
Well, thank you, Raheem, and thank you for the wonderful introduction. And I apologize for stealing your slide. Right. But I'm very excited to describe some of the work we've been doing over the past 3 years through an NSERC Alliance MyTax Accelerate project, which OWC has also been connected with, on thinking about what to do with our organic waste beyond biogas. And so broadly, our program is very, very motivated by thinking about the billions of tons of organic waste generated each year globally, and how we might upcycle those into carbon negative fuels, chemicals, and materials. And so, when we think about technologies that are well suited to this challenge, anaerobic digestion really is first of mind. And so we like anaerobic digestion. We say it's got a big mouth, right? So it can take food waste, wastewater biosolids, renewable biomasses, and convert them primarily to biogas, right? Which is a mixture of methane and carbon dioxide. But really the focus of our research program is to think what we can do beyond biogas. And so we're very interested in thinking about the chemical space and the commodity chemical space, and specifically as a starting point, these medium chain fatty acids and their derivative products that we'll talk about a little bit this morning. And so oftentimes when I tell people we want to think about beyond biogas with digestion, people say it's too complicated, the tools are too limited, and really we have just empirical methods here at play to be able to optimize these systems, so it's too challenging. And so what our program has really been doing is kind of opening up this black box, thinking more specifically about the biology. So what are the microbes that are present in these systems, and how do they interact, and what's their metabolism look like? But also really thinking about how we might develop bottom-up approaches to build microbial communities that have more altered function, and ultimately lead to higher value products besides just biogas. And so really this speaks to a need for not only new innovations at the process level, but also new innovations at the micro level or the microbe level. So when we think about products beyond biogas, I like to think about this value volume pyramid. So of course, I guess at the base of the pyramid is energy molecules. So these are things that we need in the billions of tons per year as a society. And so this is what ADI is already really quite good at, right? So we're good at taking waste materials and converting them into things like renewable natural gas, for example. But the problem is that the value of that product is inherently not very high, right? We need a lot of it, so the price is not going to be as high. On the top of the pyramid are things like pharmaceutical products. And so those things are selling for very, very high prices. You can think of in the thousands of dollars per kilogram, but we only need a little bit of them. And so when we think about addressing our climate change goals and actually dealing with all of our waste, that's not going to solve the problem either. And so what we're really thinking about in this value volume pyramid is kind of moving up one or two of the rungs to the bulk chemicals or commodity chemical space. So those are things like acids, alcohols, things that go into many different ingredients across society. Plastics and polymers, so those are things going in obviously in plastics and in our clothing, and food and feed products. And we really think that this would not only allow us to make a higher value product from anaerobic digestion, but it would also increase the widespread nature and distribution of adopting anaerobic digestion technologies, obviously here in Ontario, but also across the world. So, when we think of the products that we want to focus on, of course, as I mentioned, the chemicals and petrochemical space, replacing those with waste-derived bioproducts is certainly a big focus of ours. There's large greenhouse gas emissions associated with these hard-to-abate sectors, but we're also still certainly focused and interested in these long-haul transportation fuels. So I'm sure many of you will be familiar with things like sustainable aviation fuel, but even trucks and large shipping vessels also generally still require liquid biofuels, or liquid fuels for that matter, to be able to move things around. And so these are kind of two of the big bins that we're focused on. And so this was a really interesting paper that came out in Nature a couple of years ago talking about the refinery of the future that I think puts some of the work into context as well, especially on the products that we focus on. And so here in 2020, this is 2020, seems like just yesterday, but now that's already 6 years ago. Right, the feedstocks that we currently use in refineries, right, are oil. Primarily 86% of it is coming from oil, and other, which is largely natural gas. And we use these feedstocks to turn them into largely liquid fuels for transportation, so gasoline, diesel, and jet fuel, and also a small fraction for the chemicals and the polymers that we use in today's world. And largely, this is all, high temperature, high pressure chemical refining. In the future, right, in 2050, the vision is that a lot of the fuels and the chemicals and the polymers that we're going to be making are coming not only from CO2, so directly pulling CO2 out of the atmosphere and making our chemicals and our fuels, but directly, as you can see here, from agricultural and municipal solid waste. So about half the biomass feedstocks going into the chemicals and the polymers business is expected to come from agricultural and municipal biomass. And so, of course, these are the type of feedstocks that we on this call have a lot of expertise in handling and dealing with. And the other point I wanted to make that was really well articulated here is that a lot of the energy production in society now, and certainly moving forward, is coming from non-carbon based sources. And so we have good alternatives for making energy with low carbon sources. So wind, solar, of course, here in Ontario, hydropower is a big option. But we don't have good options to manage our carbon, right? So if we want to make the chemicals and the plastics and the materials of the world that need carbon as a feedstock, what better place to start and to look to than upcycling all the large amounts of carbon we have in our organic waste feedstocks. Okay, and so we feel that medium chain fatty acids are a really good starting point for doing this. So what are medium chain fatty acids? They're monocarboxylic acids of chain length from C6 all the way to C12. I have a range of both short chain fatty acids and some medium chain fatty acids on the slide. And really, they're a good starting point for several reasons. One, they're significantly more valuable as an end product than biogas, and so they sell for anywhere from $5 to $10 a kilogram as a bulk commodity. And they're used in a wide variety of products. So you'll find them in nutraceutical products, personal healthcare products. A big market, of course, is as animal feed. So MCFAs are actually naturally antimicrobials, and so they're being used to replace antibiotics in livestock cultivation. And you'll find them in everything like personal care products, right? My running joke's always I was working on my beard, and this is something I found at Shoppers Drug Mart. You'll find these MCFAs. So they're very valuable. They're used broadly across the chemicals industry. Those are two big selling points. Another big one is they're currently coming from really unsustainable sources. And so MCFAs currently are refined from largely palm kernel oil and a little bit from coconut as well. And you've all heard that the bad headlines, I'm sure, around palm refining. And so there's large greenhouse gas emissions, deforestation, biodiversity loss, a lot of these environmental issues that have plagued the palm industry for a long time. But also we know and understand that there's a lot of supply chain issues as well. And so you have to get harvest the trees from Southeast Asia, refine them in Europe, and then distribute them globally. And so what that ends up happening is there's a lot of price volatility and just availability of the material itself into various supply chains, which is really wreaking havoc for many people who depend on these chemicals. And so finding more locally sourced, environmentally friendly ways of making these MCFAs is a big driver for many people, end users of MCFAs. So as I mentioned, MCFAs themselves are very valuable, right? And we can make them from waste, but they can also be derivatized into a wide variety of other chemicals, really driving a biorefinery concept. So we can reduce them to their corresponding alcohols and alkanes, which have various applications, obviously in the fuel sector, but also the fuels and the fragrances. And we can also, carboxylate them, turn them into things like adipic acid, which are precursors for nylon production, for example. So there's really many different end uses and markets that these MCFAs can spill into, again, which broadens the application of this technology, not just to one chemical, but to a whole refinery idea. Okay. And so we got started on this project really thinking about how can we turn the City of Toronto's source separate organic waste into medium chain fatty acids. And so for the last 3 years, we've been going out to both Dufferin and Disco Roads organic processing facilities, collecting this nice yummy pulp at least it's yummy for the microbes anyways, and thinking about how we can develop the process technology, so that's both the fermentation methods and the extraction methods to purify out the MCFAs, as well as some of the underlying fundamental synthetic biology that's really been underpinning some of the technology and really driving the next generation of what we'll be doing with our project moving forward. So I'm happy now to pass on the torch here to Jasmine, who's going to tell you a little bit about the bioprocessing that we've been doing here in the past 3 years.
Okay, thanks, Chris. Oh, sorry.
Getting the slides up here. Third time's the lucky charm.
Okay.
There we go. Great.
There we go.
Sorry about that.
No
So I'll
problem.
just follow up on Chris's introduction. So as you mentioned, anaerobic digestion is a common waste diversion strategy that's often used to reduce the environmental impact of landfilling organic waste. And it's also a method to recover energy in the form of biogas. And so here we've shown it's a multi-step process, and it involves a diverse set of microbes, and each of them encompass different functional guilds that work together to break down the complex polymeric material in our organic waste into intermediate compounds such as acids and alcohols, and then ultimately which are converted into biogas. But by driving the microbial processes away from methane production, we have the potential to derive even higher value chemicals such as medium chain fatty acids. And we can do this by using the intermediates of anaerobic digestion, so notably lactate and ethanol. So as Chris mentioned, MCFAs are an attractive product, not only because they have a higher market price than biogas, but they're also used in a wide variety of industrial and agricultural applications. And ultimately, what we want to do is produce MCFAs locally and more sustainably using organic waste. However, a major challenge to consider when working with microbial communities and organic waste is byproduct formation from competing pathways, which can limit the production rates of our target product. And this is something that we'll address in our presentation. So since we wanted to produce MCFAs from real waste streams, we collected feedstock from 2 different full-scale anaerobic digestion facilities in the city of Toronto. So one is the Dufferin Organics Processing Facility, and the other is Disco Road. And both of these facilities collect and treat source-separated organic waste through the municipal green bin program. And at these facilities, they will basically collect the solid SSO and pre-process it to remove inorganic material, and then combine it with processed water to form a pulp or a slurry, as you can see highlighted in red. And this is what is fed into digesters to produce biogas. And for our study, what we are using is the SSO pulp as our feedstock. So the overall aim of our study is to demonstrate long-term stable MCFA production and recovery from SSO in a continuous bench-scale bioreactor. So in this study, we used an open culture fermentation approach where we're basically just adjusting the process parameters of our system to enrich the microbes that are responsible for MCFA production in the reactor. And lactate is one of the key electron donors for this entire process, and its availability in the SSO feedstock significantly influences the bioreactor performance, which I'll get into. And we operated this reactor for around 2.5 years, and it was continuously being fed the SSO pulp. So initially from the Dufferin facility, and then partway through, we switched to using SSO from the Disco Road facility. So in this long-term study, we looked at the impact of many different operational parameters on bioreactor performance. So we tested increasing the solids retention time from 8 to 12 days. We also tested facility-specific and seasonal variability of the SSO feedstock. And then finally, we looked at adding inline product extraction, and this allowed us to actually produce our MCFAs in their final oil form. And we also looked at investigating how the microbial communities in the feed samples and in the reactor were changing over time in response to all of the process changes that we were making. And so we'll share some of our key findings that we found. So first we'll look at the variability of the SSO feedstock itself. So in this figure, we have the concentration profiles of key substrates in the SSO from the Dufferin and Disco Roads facilities over time. And the timeline is sort of split into different periods reflecting changes in the outdoor seasonal temperature when the SSO was actually collected from the facilities. So in blue, we have like a colder period, and red, we have warmer periods. And what we basically found is that the SSO samples initially from the Dufferin facility were found to be generally pretty stable over different seasons. So for example, the main electron donor for a processed lactate generally remained pretty high in a concentration, so around 10 to 20 grams of COD per liter. But in contrast, the SSO samples from Disco Road were significantly impacted by the changes in outdoor temperatures. So in the warmer periods, we found that the lactate levels are lower and sometimes even like 0 grams per liter due to the fact that they were being consumed for short chain fatty acid production before we even collected the pulp from the facilities. And we see that in the colder periods, the lactate levels were higher, and this is presumably because of the slower rates of SSO degradation prior to collection. So now we'll look at how this feedstock variability that I just showed is influencing the MCFA production in our reactor by looking at the product profile of the short chain and medium chain fatty acids in the bioreactor. So in this figure, the dashed lines are representing feeding rates and the solid lines are representing effluent rates leaving the reactor. So initially, what we see is that when we're using SSO from Dufferin, which had better lactate preservation, as I mentioned, the MCFA production is quite stable, and it's dominated by even chain MCFAs, so C6. However, when we switch to using SSO from Disco Road during a warmer period, we see that there is a higher feeding rate of C3, which resulted in production of an odd chain fatty acid, such as C5 and C7, and overall a lower production of even chain MCFAs. And once the SSO from the colder period was introduced, the even chain MCFA levels recovered back to the previous period, and the overall MCFA production rate improved. So overall, what these results are showing is that the quality of the SSO feedstock, which can be like temperature driven, it impacts the MCFA selectivity and also the product profiles that we see in our reactor. So when we were thinking about the reasons for these observations, we think that there are two potential sources of the SSO variability between the two facilities. So the primary one is the transit time between the curbside collection of the SSO waste from our homes to the tipping floor of the facilities. So we think that the differences in residence time at the transfer stations between these 2 points could explain the variability of the SSO degradation beyond lactate. And another source is recycling processed water into the SSO pre-processing step from digester effluent streams, and this could introduce variability in the microbial composition of the SSO itself. And later on, Diana will show further evidence that we've provided from our data that also suggests this might be the case. And altogether, these results are highlighting the unique challenges of working with real organic waste over a long period of time. And now I will pass it off to Ethan, who will discuss the engineering of microbial communities.
Hey everyone, can you see my screen?
Looks good.
Great.
Looks good.
So my name's Ethan, and I'm a PhD student in Professor Lawson's lab. And Jasmine described a little bit about how the bioprocess, we're doing research to improve MCFA productivity. But on my research, I'm looking into how we can actually modify the microbes themselves to increase productivity. And we call this approach both synthetic biology and metabolic engineering. So as Professor Lawson described, there's an opportunity to improve the product spectrum of anaerobic digestion through chain elongation. MCFAs themselves have a huge market size and service a lot of different industries, including food and feed, but we can also access other bioproducts, such as fatty alcohols, diacids, and diols, which can be used to service hard to decarbonize sectors like sustainable aviation, long-haul transport, and as well as industrial chemicals. However, in order to accomplish this vision, we need a level of control on the actual bacteria that reside in these communities to enable the production of these different compounds. And this is where my project comes into play. And I will discuss throughout this presentation how we developed a method to deliver engineered genetic code into the chain elongating bacteria responsible for MCFA production. And I'll show that we have demonstrated this pipeline in a chain elongating bacterium called Clostridium cliveri, and also about our future directions and how we want to improve chain elongation. So to start off, I wanted to introduce an analogy to help explain synthetic biology and metabolic engineering approaches. So much like how we use CDs, we insert them into stereos to play our different music, we use things called plasmids that contain the biological data encoded in DNA, and we deliver them into the bacteria, which is the hardware. And by doing this, we are able to affect the function of the bacteria. And for example, we can access different bioproducts. And with a whole suite of different plasmids, we can affect the function of these bacteria to access a wide range of different chemicals. So this is where my project started, and I will introduce now how we kind of approach this delivery pipeline. So as a context, bacteria have an innate immunity system against this external genetic DNA. So it's called the restriction modification system. And how this works is that if we try to input plasmids into bacteria that have not been engineered before, they have enzymes in the cell that will break down the genetic code, which then prevents the genetic code from functioning. So what I've done in my project is I've developed a method to protect the DNA before it is introduced into the cell. And we do this by methylating the DNA, which is just a modification of the genetic code before we deliver it. So how this works is that we take an enzyme called a methylase, we treat the DNA before it is delivered to the cell, and once we introduce it, it is protected from degradation. What this now allows is that the genetic code can now persist in the cell. Therefore, it remains functional and can be used to direct the function of the bacteria. So this is a schematic of how this works, is that we take the unmethylated plasmid DNA, the genetic code, and we use a helper strain of E. coli that does the methylation that protects the DNA and then also delivers that into the cell. What I'm showing here now is data showing that without this pre-protection with methylation, we obtain no colonies or no growth of our engineered bacteria. But upon introducing methylation, we are able to obtain very high efficiency recovery of engineered bacteria. And then now what I'm showing here is that the same data is that we were able to replicate this over many different plasmids that all have different functions. And when it's unmethylated for all plasmids, there is no effective recovery. But when we methylate it, we are able to get a lot of these different engineered bacteria. So this was the first piece of enabling metabolic engineering in these strains. Now, the second piece was to show that we can actually change the function of the bacteria. So using this pipeline, I introduced plasmid DNA or genetic code that will allow us to detect through fluorescence modification of the bacteria. So how this works is that the plasmid that we introduce has the genetic code that encodes for a particular protein that will glow under UV light. So now what I'm showing here is that when we provide this plasmid DNA with this particular enzyme, this protein, we can show that we have strong fluorescence from the modified strains and no fluorescence from the natural strains. So the figure here is showing the level of fluorescence we're obtaining from the modified strains. So now this is proof that we can functionally change the bacteria Clostridium clavirii, and we are looking to apply this into now changing it to more productive efforts for MCFA bioprocess technology. First, we did report this work in ACS Synthetic Biology. So if you're interested in diving deeper into the genetic engineering pipeline that we developed, the paper is available online. But now the interesting part is we're trying to use this tool to actually modify the function of Clostridium clavirii. What I'm showing here is a metabolic map of the reactions that this strain conducts. For example, it takes ethanol and it will convert it naturally into these medium chain fatty acids, including butyrate, hexanoate, and octanoate. But if we want to access these other bioproducts like longer chain fatty acids or fatty alcohols, for example, which are useful in long-haul transportation fuels, we need to be able to add functions. For example, adding the functions required to convert the cellular intermediates into the fatty alcohol, but we also need to be able to remove functions and reactions to fine-tune the metabolism so that we increase the yield, growth rate, and the chain length of these bioproducts. So this is kind of the next step that we want to use our genetic engineering pipeline for. So coming in the next coming year, I'm working with Ian Goyce, who is also in Professor Lawson's lab. He's conducting protein engineering efforts in order to access longer chain fatty acids. So how this works is that we also use genetic engineering to modify the enzymes themselves. Enzymes are the catalysts that turn the substrates like ethanol or lactate into the fatty acids. And we want to do this because with increasing chain length, we are able to access increasing market price. Furthermore, I'm also working with Abi Ali Badani, who is working on doing some metabolic engineering to enable product diversification. So this will allow us to access compounds like methyl ketones, fatty alcohols, alkanes, diacids, and diols, which not only are have larger market volumes, so there's a larger demand for them, but also service things that anaerobic digestion are not able to do yet. So this will allow us to access markets like aviation fuel, chemical processing, and this will really increase the impact and potential of anaerobic digestion for sustainable production. So in summary, I've developed a method to deliver genetic code into chain elongating bacteria. I demonstrated that it worked with the fluorescence signal, and now we're working on enhanced MCFA production and product diversification. And overall, how we see this contributing to MCFA bioprocess technology is that with the engineered chain elongators, we will re-input them into the community in the bioreactors. And with that, they're able to then produce these wide range of different bioproducts. All right, so that's my presentation. I'd like to hand it off to Dr. Jay Werber.
All right, hi everyone. So my name is Jay Werber. I am the PI of the Advanced Membranes Lab here at U of T. Sorry, let me just try and maximize this again. I just want a brief intro of my lab in terms of what we do. So we work on developing membrane materials and membrane processes to address major sustainability challenges, largely through addressing the energy intensity of industrial separations, which consume about 12% of global energy. We work on a variety of different membrane applications, including reverse osmosis for desalination, electrodialysis for desalination, and also for selective ion separations in the hydrometallurgy space. But what I'm going to talk about today is how we've been working with Professor Lawson's team on the purification of these bio-derived chemicals, these medium chain fatty acids. So in terms of developing a purification process for these MCFAs, the first thing we have to keep in mind is what is the final product. And there's two possible options. The first one is to produce a mixed MCFA oil, something like C6 to C12. Ideally, it would be in the neutral acidic form. This is the higher value form. or we could produce sort of isolated, purified chemicals that could be used for particular purposes. Of course, from a desired process, we want it to be low cost. We want it to be low carbon footprint. In other words, we want this to be run on electricity, because electricity can be renewable instead of fossil fuel-based heat sources. We want to use inline MCFA removal to decrease the toxicity to the cells, and this can increase the production of these MCFAs, which will be discussed in a bit. We would also want it to be solvent-free, so that solvents won't get sort of entrained in the bioreactor and impact growth. And of course, we want this process to be robust and easy to run. So our initial focus is on producing a mixed MCFA oil. We are thinking about possible processes that would isolate purified MCFAs, but this may require several distillation steps, which would be costly and quite energy intensive. There could be other ways too, but distillation would be the most likely source. So in terms of distillation, this is actually what a traditional chemical engineer would propose to purify MCFAs from a bioreactor broth. And so, for example, NROL has been looking into various carboxylic acid productions from anaerobic digestion, and they've considered this process where they would use a extraction step, which they're showing as the membrane contactor. Sorry, let me get my laser pointer. So this membrane contactor is a liquid-liquid extraction step. So this is a conventional using a solvent. They propose using mineral oil or various more extracting oils, and extracting the MCFAs into the oil loop, and then using distillation to recover both the MCFAs as the volatile product, and then the solvent as the low boiling product on the bottom. And so this works by selectively extracting the more hydrophobic species. The downside is that you have sort of a solvent, and you have high energy needs and relatively high capital costs. And so the process, the overall process that we've been considering for this application is from the bioreactor, we would use a submerged membrane to keep back the solids. The solids-free solution would then go through this PDMS membrane step, which is a membrane-based extraction process that uses polymeric membranes in a completely solvent-free format. This would extract the MCFAs into a high pH solution as carboxylates. So these carboxylates would then go to an acidification tank where they get acidified using something like sulfuric acid. These MCFAs are highly soluble when they're in their ionic high pH form, but when they're brought to low pH, where they're now neutral, they're relatively hydrophobic, they'll crash out of solution and form a MCFA oil. So this is your product. The residual stream is this salt solution that is at low pH, and we want to basically use a process called bipolar membrane electrodialysis to split that salt and produce an acid stream and a sodium hydroxide stream, which basically recirculates the chemicals, allowing for sort of a circular process to be used. So I'm going to focus on the silicone membrane part, which is this robust solvent-free extraction process that produces the MCFA solution in the salt form. And this process is using, again, these membranes called PDMS, or polydimethylsiloxane, which you will know as a silicone. And so the nice thing about silicones is that they have a lot of molecular movement at the molecular scale. This can lead to really high permeabilities of species going through them. And so the way it looks is that on the left, we have a solution from the bioreactor that's around pH 5. We're going to have a mix of the acids, both in terms of the types of species, but also in terms of their ionization state. So some of them are going to be in this acidic form, and some of them are going to be in the carboxylate form. Because the silicone membrane is really hydrophobic, only the non-ionized acidic form can partition in. And so what happens is they diffuse in, they partition in the acidic form, diffuse down their concentration gradient, and then pop out on the other side. Because the other side is really high pH, that acid-base reaction is really quick, causes them to become this sodium carboxylate immediately, and this provides the driving force for permeation. We think of permeation through a membrane like this in my world as being something called a solution diffusion type mechanism, where the permeability is related to the sorption coefficient, basically how much the species will dissolve into the membrane, times the diffusivity, the diffusion coefficient for the species in that matrix. Basically, how fast can something move if you have a certain amount of driving force. And so we would do these diffusion cell diffusion cell tests, where we would have a mixed solution of the various carboxylic acids going from C2 up to C8. You can see that the C8 is being depleted really fast in the feed stream and is popping into the strip side extremely fast. And this suggests, based on this data, that the C8 is going through the membrane faster than, for example, the C2, which is much, much slower. And so using this data, we can extract what's called the permeability. And this is the intrinsic ability of this material, silicone, to transport these various chemicals through them. And what you can see is that we have this selectivity towards the more hydrophobic species. So the fattier species dissolve into the membrane more. So this is the sorption coefficient. They dissolve into the membrane more and then diffuse through at about the same rate. And so this ends up resulting in the much, much higher permeability. And so this is great. This suggests that these silicone membranes enable rapid, selective, and solvent-free recovery of these medium chain fatty acids into an alkaline solution. And this is a solubility-based selectivity that is nearly identical to extraction with mineral oil. In other words, we have this solid, relatively low-cost process that uses no solvents at all, that is able to produce similar performance as the traditional liquid-liquid extraction technique. So as a last thing, we looked into what commercial membranes were available to try and scale this up. The main one that was available on the market is something called a hollow fiber silicone membranes. These are dense tubes with silicone walls. And so these are commercially available from at least 2 suppliers. We tested one of them, and we found that indeed it still has really good selectivity for the C8s over the C2s. The downside, though, was the permeability was quite a bit lower, and we're still not really sure why, but probably something to do with the material characteristics of this membrane. So we have to look for other paths for scale up. This is something that Renzo is going to discuss in a little bit. But for now, we've been using this hollow fiber module in our lab scale setups, which will be discussed soon. And that's it. Thank you. So I'm going to be moving the slides for Diana. Oh,
just a minute's gonna move slides.
Okay, great.
Okay, so I'll be continuing our bioreactor presentation, specifically looking at the impact of inline MCFA extraction on performance metrics, and also we'll be looking at microbial community profiles. So MCFAs are known to be toxic to microbial activity, with previously reported toxicity levels of around 7 millimolar. And before we started extraction, our maximum MCFA titer was around 2-fold larger, suggesting that MCFA production was already limited by inhibitory effects. So therefore, our motivation for implementing inline extraction was to reduce MCFA toxicity in the bioreactor and thereby produce the production metrics. To achieve that, we had solid-liquid separation system in the bioreactor, as Professor Jay mentioned, where the permeate loop from the bioreactor was recirculated through the PDMS membranes, and on the other side, alkaline solution was recirculated and continuously extracting MCFAs. And once that alkaline solution was concentrated, we were able to phase separate MCFAs in their final product form as oil via simple acidification. And an advantage of using these PDMS membranes is that they're solvent-free and extractant-free compared to conventional liquid-liquid extraction processes. So now we'll take a bit of closer look how MCFAs are extracted from the bioreactor. Approximately half of MCFAs exist in their protonated or undissociated form at pH of 5. Which is the toxic form of MCFAs to the microbial activity. So we want to extract continuously them as they travel with the permeate loop. They come into contact with alkaline solution, here sodium hydroxide, and are extracted as MCFA salt, where extraction is driven by a MCFA concentration gradient on the membrane layer, which is created by pH difference between the bioreactor at pH of 5 and alkaline solution at pH of 9. And again, once this alkaline solution is concentrated, it can be phase separated to produce our MCFA oil product. So next we'll take a look at the bioreactor performance with and without the extraction during colder periods when we're using non-degraded source-separated organic waste as feedstock. So we observed that the MCFA production increased, specifically hexanoic, C6, and octanoic acids, as well as the MCFA yield improved by around twofold. Additionally, we saw decreased production of odd-chain volatile fatty acids, suggesting that with inline extraction, there was more selectivity to even-chain elongation. As we transition to using warmer feedstock, we see that the MCFA production decreased, and similarly to the previous warmer period, we saw increased feeding rates of propanoic acids and decreased feeding rates of lactate, which subsequently limited our MCFA production. And the yield also became similar to the warmer periods. So overall, this results again highlight that once the feedstock is degraded past lactate, MCFA production is limited on the substrate level. Next, we'll take a look at the microbial community profile in the feedstock from two different organics facilities, which nicely complemented our chemical data. First, we see that in Dufferin feedstock, primarily lactic acid bacteria in the genus of Lactobacillus dominated the community, which explains the high lactic acid concentration we observed in that feedstock. When we look at the feedstock community profile in Disco Road, we see that on the days where lactate was preserved, there's a high relative abundance of another lactic acid bacteria, Streptococcus, and on the warmer days, as we've just seen, where lactate was more degraded and short-chain fatty acids concentrations increased, we see increased relative abundance of short-chain fatty acids producers, including Prevotella and Megasphaera, both of which are associated with propionate production. And altogether, these results again highlight that there are likely facility-specific processing conditions that resulting in this different microbial community profiles, and as just mentioned, it might be from waste collection systems or different residence time of the waste. And next, we'll take a look at the microbial community profile now in the bioreactor across the operational period. When we're using different feedstock, we see two dominating bacteria. First, lactic acid bacteria, Lactobacillus, and our second main chain elongating bacteria, Eubacterium, or now called Peptonella actinoica. And as we recall, during the operation with different feedstock, we demonstrated stable MCFA production. And when we look at the community profile with Disco Road, we see that on the days where we demonstrated increased MCFA production, we see another putative chain elongating bacteria, Pseudorimibacter, and on days where we had decreased MCFA production and increased levels of short-chain fatty acids in the feedstock, we see again those putative short-chain fatty acids producers. And additionally, with Disco Road feedstock, we see putative butyrate producer that is present across all time points, which correlates with our chemical data where we see increased butyrate production. And overall, these results highlight again that during warmer periods, those short-chain fatty acids potentially competed with chain elongating bacteria, resulting in our decreased performance, which motivates the next study which is to use synthetic microbial community consortia to improve selectivity towards MCFA production. Lastly, we'll look at the composition of our final product, which is MCFA oil. As mentioned previously, we are extracting MCFAs in this alkaline solution selectively, as you see in the first panel, where mainly we have C6, C7, and C8. Once this alkaline solution is concentrated, we can phase separate them via acidification, and we see that the majority of MCFAs end up in the oil phase, which can be further improved if we let the alkaline solution concentrate to higher concentrations. And finally, our MCFA oil was found to be quite rich in terms of gram of MCFA per gram of oil. So these results highlight that with simple acidification, we are able to produce our final product MCFA oil without needing additional downstream process units. And lastly, we wanted to highlight that we recently submitted our paper on the long-term production and recovery of medium-chain carboxylates from source-separated organics. And now I'll pass the presentation to Renzo.
All right. Can everyone see my screen?
Yep, looks good.
Perfect. Thank you. Thank you, Deanna. And yeah, my name is Renzo. I'm a master's student working with Professor Jay Werber. And a big portion of my project has sort of been focusing, again, on all of the downstream processing that happens in the MCFA production line. So a lot of my focus for this part of the presentation is going to be our efforts in scaling up the inline MCFA extraction. As Professor Werber mentioned, we have shown that PDMS, both in a freestanding flat sheet geometry and also in a hollow fiber PDMS geometry, could be used for the extraction of the MCFAs. So just to reiterate what Jay brought up previously, what we looked at in our paper was the permeability coefficients of both the freestanding PDMS and also the hollow fiber membranes. And as you can see here, yes, we do get great selectivities using both of these membranes, but what we do see is about a 50-fold, greater than 50-fold decrease in permeability once we move to this hollow fiber geometry. And what we sort of suspect is that the decrease in the performance of the membranes could be due to perhaps additional fillers in the membranes that make them more mechanically robust. Jay might have mentioned this, but the typical application of PDMS membranes, especially in the hollow fiber geometry, are for gas separation applications. And in those applications, you can imagine that these membranes have to withstand really high pressure gradients. So in order to make them more mechanically stable, they do have to adjust the PDMS chemistry. But for us, that's the worst case because we also have the transport of the MCFAs affected as well. So an alternative for PDMS membrane scaling up is what we call PDMS thin film composite or TFC membranes. So instead of sacrificing the PDMS chemistry, what we do now is that we can take what's called a support membrane, which is a porous membrane as shown in the gray, in this gray area over here. And what we can do is we can cast a PDMS layer on top of this support membrane in order to provide mechanical stability, again, without affecting the chemistry of the PDMS. So sort of imagining how this would look like at scale up, comparing that to how hollow fibers would look, we would have our MCFA rich solution passing through what's called the lumen side or the inside of the hollow fibers, and then our extract solution circulating countercurrent to the MCFA solution on the outside or the shell side of the hollow fiber membranes. But with PDMS TFC membranes, what we can imagine is what we call a plate and frame module. So just from this schematic here, as you can see, first of all, we have both our MCFA feed solution and our alkaline extract solution. And if you look at the stack carefully, you can see that the membranes are oriented so that the flow path of the extract solution is only facing the porous support layer on the sides of the membranes, and the MCFA feed solution is facing the PDMS selective layer side of the solution. So altogether, this could be compacted into what we call one stack of membranes, and we would have these solutions again countercurrent, and we'd be able to achieve that selective extraction of the MCFAs by arranging the membranes in this sort of module orientation. So luckily, a vendor has given us some PDMS TFC membranes to test. Again, for this vendor, their main application of these TFCs was for gas separations, but we had about five samples of these PDMS TFC membranes, which we characterized by cross-sectional SEM. So just as an example as to how these membranes could look in terms of their design, I just wanted to show you guys these 5 different types of membranes here. And as you can see, the type of support that you use can vary greatly. So if we were looking at M1 over here, let's say, these pores on the porous support look like, they're very big and open finger-like pores. But then if we look at M2 to M4, some of these support layers can be more dense, less porous, right, compared to M1. So in terms of membrane design, one of the big questions is how does the support layer affect the transport of the MCFAs then, since now we have this additional layer of resistance, we call it. And just in general, when we were doing our initial testing with these membranes, we found that M0 performed the best. So later when I show you some preliminary results, that's the one we'll be focusing on. And yeah, as I mentioned, with this additional support layer, we also have, again, this additional resistance, which could affect the transport of the MCFAs. And as I showed in the previous slide, depending on the type of support you use, that could potentially have a larger impact on transport than we think. So one of the ways that we can sort of address this challenge is if we were to buffer the alkaline extract solution to maintain that pH driving force, we can sort of try to mitigate the effects of the support layer and try to get better extraction, better transport of the MCFAs. So like what we did for our previous study, we could determine the transport metrics of the MCFAs using diffusion cell tests. Now, in a lot of my preliminary work, I've been using a mixture of C2 to C8 MCFAs, where we hold the feed we adjust the feed solution to a pH of 5, and then on the alkaline extract solution, the sort of 2 conditions that we wanted to test is one condition where we did a pH 13 sodium hydroxide extract solution with no buffer, or a pH 10 extract solution with 100 millimolar sodium bicarbonate to act as a buffer. So again, these membranes that we tested were the M0 membrane, as I showed previously. And what you can see from here, so here I'm showing the percent extracted of MCFAs in the extract solution. So these are just time courses over the span of about 4 days. And as you can see, what we have for both of these cases is that we have the extraction of C6 and C8, right? That's what we're sort of focused on. But after about 24 hours, what we see is that we sort of lose our extraction efficiency when we look at the solution, the alkaline solution where we don't have a buffer, so the one on the left. Whereas the one with the buffer, we can see that we can still maintain the extraction efficiency of our extract solution when we have a buffer. And sorry. And another thing that I wanted to show as well is their permeance and their permeance selectivity. So the permeance is essentially just a measure of how well the material performs. It's the mass transfer coefficient of the MCFAs. And as you can see, just by looking at the permeances and the permeance selectivity, we get better selectivities, more preferential selectivities towards the longer chain MCFAs when we have a buffered extract solution. And sort of moving forward in this scope of the project, again, since we have this additional support layer, what I've been working on is developing a 1D analytical model to sort of answer some of the questions that we might have in terms of membrane design. So things like, how does the PDMS thickness affect the MCFA extraction? How do certain characteristics of the support layer affect MCFA extraction, right? So that's one thing in terms of thinking about scaling up and how we can make these membranes. That's one thing that I've been working on. But another thing that I've also been working on, sort of connected to this modeling stuff, is making our own PDMS TFC membranes in the lab and using those to validate this model. And again, use the model for membrane design and scaling up. So the reason why we want to use membranes the reason why we have to use membranes that we make in lab is because we know exactly what the PDMS chemistry is and exactly what the support layer characteristics are to help us validate the model. Obviously, with the ones that we get from these vendors, we're not sure exactly the chemistries and the characteristics of the materials that they use. And then here I just show some of the work that I've been doing as well in making these membranes, which so far have been successful. And yeah, with that, I'll pass it back to Chris for wrapping up.
Great. Thank you, Renzo. If you could advance to the next slide, that'd be great. Awesome. Okay, well, thanks everyone for staying along through the journey here with us this afternoon. And so I just wanted to highlight quickly some of the key accomplishments of the work we've done here over the past 3 years. And so there's really 3 main things we've focused on. And so at the microbe level, we've been the first group to really develop new genetic tools for some of these weird anaerobic microbes that naturally make medium chain fatty acids. And that's been very, very useful because it's allowing us to not only optimize MCFA yields and productivity at the molecular level, but it's also allowing us to elongate MCFA's other products, alcohols, diols, and diacids. Then on the process side, as you've seen, we've been able to demonstrate long-term production of MCFAs both in a bioreactor using real organic waste from the city of Toronto, and also developed PDMS membrane systems to recover the MCFAs as an actual free acid. And so this has been remarkable work by our trainees, and what we're focused on now is moving toward what you've heard a couple times pop up as synthetic microbial communities. And so on some of those really colorful slides, Diana had showed you with all the colorful bars and the funny Latin names for microbes. What we've been doing outside of this project is actually isolating a lot of those microbes in high throughput and genetically modifying some of them. And so we now have a synthetic community that we're trialing with SSO in the same process to see the MCFA production gains that we have. So stay tuned for updates on that. Next slide, please, Renzo. And so I want to give some really, really big shout outs to the people who have allowed this program to run for the last 3 years. And certainly that starts with our Waste to Chemical Alliance. And so we've had deep partnership engagement from the onset of this program. And I think that really is one of the key reasons why it's been so successful. So we've worked with the City of Toronto, Ontario Clean Water Agency, Veolia, of course, the Ontario Water Consortium, as well as Envera and CBS Bio platforms. And so really these stakeholders span the so-called waste to chemical supply chain, right from the feedstock side to the technology developers to the end products, who are people who are bringing these products to market. And so our interactions with them has really helped define the scope of work that we focus on and shape the science. Of course, I'd say the most important outcome of the project and the biggest piece of acknowledgement that I want to give is to the trainees who you've heard talk to today, and of course some of the trainees that you haven't been able to weren't able to present today, but have also been really key in driving the work. And so I think from Jay and I's perspective, anyways, that's the most important outcome, is really developing the future talent that's going to really implement these changes in the water sector. And so we're here for, I guess, another 20 minutes, Raheem, right, to answer folks' questions on the call, and so happy to field those questions and pass them over to some of the people you've heard speak today as well.
Well, fantastic. Thank you, Chris. Thank you to all the presenters. So yeah, we can maybe have the presenters come on screen with your cameras.
Yes, please.
And and if there are any questions, just raise your hands, and then we can go through them. You can the attendees can also turn on their cameras now and unmute themselves. So yeah, feel free to put your hand up with any questions, or to drop them in the chat if you prefer to do it that way. And while the attendees here are thinking it over, perhaps Chris or Jay, I mean, that's a lot of fantastic work from advanced biology through to material science. You know, what's and lots of good results, and you know, obviously lots of IP that's being generated for you and the team. I mean, what's your most optimistic or your most hopeful outcome that you want to see from all of this hard work, whether it's, you know, at the end of this particular NSERC project, or looking to the next one, or even longer term?
Yeah, I can take that on. And so of course, you know, science is good. Getting papers out is good, and we've been quite successful with that as well. But ultimately, what we want to do is we want to see this technology enter the market and be a real technology and a real option to municipalities, to industrial waste sources, for being able to extract as much value out of their waste, because we really do see, you know, waste not as a waste, but just as a future feedstock for, not of course for biogas production. I don't think that's going to go away, but certainly also for recovery of higher value chemicals. And so next steps, what we're doing is looking at pilot plants. So we have some ongoing pilot discussions with some of the work to really scale it up and take it to the next level, both on the municipal side, but also on the industrial side. So looking at industrial waste streams as well. And so yeah, the biggest thing I look forward to, I won't speak for everybody, is really commercializing the technology, seeing it come to market, working with everyone on this call to really embrace innovation in the water sector. I think across all industries, you know, from pharma to chemicals to waste, what we're proposing here is really a brand new supply chain. And so because of that, nobody owns it, right? You talk to a waste company and they're like, well, I don't do chemicals. You talk to a chemical company, well, I don't do waste. You talk to a fuels company, well, I don't do chemicals. And so this doesn't exist yet. And so I think it really is going to depend on the entire industry to come together. And I really think it's the waste industry and the water industry that's going to lead this to make innovations like this come real, because you know, we know the most about waste handling and waste as a feedstock. So I think that's where it starts.
Thank you.
I talk too much, so I'm happy to get questions from the audience.
Any hands here for any questions here from the audience? You talked about some piloting and sort of as your next steps, Chris. What would your ask be from the audience? Are there any challenges that you're facing? Is there anything that you might request from those online here?
I would flip it around, actually. I would say to those online, I would say, how might a technology like this solve a problem that you're having? Or maybe if you have a specific problem, either with anaerobic digestion technologies you have, or with waste, your management of solids, for example, if you're a municipality, what are the problems that keep you up at night, and how can we help solve your problems? I think that's the question I would ask.
Yeah, great. Any responses from our audience on that question from Chris? You think keeping folks awake at night?
Sasha.
I guess I can go.
Oh,
Go
yeah.
ahead, Chandra.
Well, the big one is like, how do we financially deal with our waste? That, we only have so many outlets, and the way municipalities are set up, we're very limited in how we can gain revenue. So, providing that kind of industrial symbiosis lens of, hey, wastewater treatment, waste management aren't waste, they're actually a resource, trying to frame that for both taxpayers and those who regulate us. How do we provide this as a solution?
That's a great question. And so as we embark deeper into commercializing this technology, the financial piece is there, right? So we can put our process metrics into a techno-economic model and demonstrate that the financial return is actually sufficient to actually pay back these facilities at quite reasonable payback period. So less than 5 years, for example. I think not the problem, but the challenge, and where the community needs to come together, is really addressing all the risk pieces, because this is new, right? And so really demonstrating that, there's, if we're making a product, a chemical, some of it goes just into industrial applications, and that's fine, but some of it's, maybe going into an animal feed product. And so is there a yuck factor there? This is new. Like, are there concerns about that? So that's one piece. And then really, again, it's just building out the supply chain, because this doesn't exist. And so having a unified voice saying, this is an opportunity, we want to work together. We want to build an alliance, not only within the waste industry, but across the aisle to our friends in the chemical sector, for example. And really, building out that relationship, I think is absolutely critical. Yeah. And not one overnight. It's something that we have to continually work at, right? To really make sure people understand and appreciate the value in the waste that we have. Yeah. Thank you.
Thanks, Chris.
Sasha.
Great. Well, first, just like to compliment everyone. This is one of the best webinars. The flow was perfect. Everybody had a great presentation. It was seamless. Very, very well done. And then maybe just to answer, Christopher, your question, and one that I was thinking during it, is one of the challenges with the concept of chemical, and one of the advantages of, say, I would say RNG specifically, but there's other, like the SAFs of the world, which you mentioned, are you can very easily monetize the environmental attributes associated with RNG. It becomes a very, easy thing to do carbon intensity scores, use some of the programs that are available throughout Europe and Canada and elsewhere in the world. I guess my question boils down to, have you looked at monetizing the chemical offset when you're using these alcohols or oil products as an alternative to fossil-based oil products? Or is it just that these are all high-value products, and if you can make those high-value products, that's enough on its own, and you don't even need to think about the environmental attributes?
Yeah, that's an excellent question, Sasha. Thank you. And so I think the advantage of what we're doing is that the techno-economics don't rely on a green premium for the product. So we can make the product at a cost competitive price point without any additional government incentives to deliver to make the product at cost, right? I think the advantage as well, though, is we also have a major impact on greenhouse gas emissions, for example, from, say, the palm industry. And so we can demonstrate that. from a life cycle assessment perspective, where the MCFAs that we can bring into the oleochemicals market and the offset of palm-derived equivalents is dramatic in terms of greenhouse gas emissions. So should governments actually regulate chemicals, not just the fuels that are currently being regulated, this would be a floodgate for an opportunity, I would say, for waste in general.
Very good.
So
Great.
So big opportunity, and I do think that coming down the pipeline, we will see regulations, much like what we've seen for RNG coming down the pipeline for the chemicals industry. Just haven't caught up yet to the fuel space.
I have one more question if there's nobody
ahead,
else raising their hand or jumping in. I thought it was really interesting how you compared Disco Road to Dufferin facility and how there was pretty substantial differences. Like, I suppose the time to transit is one concept, but in reality, it's the same trash collected across the broader city. Is there anything more that you need to do with that work, or is it that impactful? Like, were those, I think there were lactic acids, were they that important, or like were they deal breakers? The fact that the Disco facility had lower lactic acid, did that create a deal breaker, or what? Can you comment any more on that?
Yeah, that's a great question. We think that the differences in feedstock or the fluctuations certainly arise from transit time. So, what we think happened is that there was a lot more direct curb to tipping floor transit of the SSO happening at Dufferin, whereas at Disco there was more residence time between transfer stations and other collection points. And so in the summertime, when it's really hot, that difference really degraded the food waste beyond lactate entering the facility. And so in terms of process metrics, overall, our average MCFA yield was about 0.24 gram per gram. So overall, that's fine. And from our techno-economics, that's still quite compelling. The main question would be, obviously, if you're in the business of making MCFAs in the summertime, when you have butyrate production, that's something else we can still recover and sell as a product. It's maybe about half the price of octanoic acid, but certainly still a value. And so we're looking at also recovering the butyrate, but also co-digesting in facilities like in the summertime at Disco, where you have a large pool of short chain fatty acids, and you could bring, for example, industrial sources of waste that have high lactate or high ethanol. Certainly in the dairy and the brewing industry, there's lots of point sources, and doing a co-digestion scenario really to bring up the electron donor and allow those shorter chain fatty acids to then be elongated to the MCFAs. So there's lots of things that could be done on the process level. On the microbial level, we're doing a lot of work on optimizing the community. And so we've talked a lot about lactate here. One of the really cool microbes that we've worked with is actually not only a lactate-based chain elongator, but also directly takes proteins and amino acids to MCFAs. And so that's something that we're now just testing in the lab in our similar setup. And we think that will dramatically help increase MCFAs during kind of these hot season peaks we see in Disco. What we're learning as we study the biology of these organisms, and it's a quite an understudied biology, is that there's a lot more flexibility in the substrates that can come into some of these particular microbes. And so we've been able to find some of these key organisms, and now we're working on strategies to stabilize them in our reactors. So, yeah.
Very good. Thanks very much. Appreciate the time.
Thank you. Appreciate the questions.
All right. Well, Chris and team, I don't see any other hands, so unless we want to wait just one more minute in case somebody wants to get a final question in.
Final word.
Yeah, final word. That doesn't look like anybody wants to take us up on that opportunity. So I think with that, Chris and Jay and team, thank you very much. Presentations were fantastic. Your excellent work that you're doing. I don't know, Jay, do you want to, or Chris, do you want to have any last comments here, and then we
I
can.
was I was going to say thanks everyone for listening, and really again a big shout out to the trainees here, Renzo, Diana, Jasmine, Ethan. These are the people who are doing all the hard work, and as you can see, they're able to pull not only the science together, but also communicate the science in a very coherent, clear way. So just a big shout out to the trainees that are again going to be leading this effort into the future. So thanks everybody so much, and please feel free to reach out if you want to follow up. So happy to chat.
Wonderful. Well, thank you, team. Thank you, all the attendees.
Thanks, Raheem.
All right.
Take care.
Thanks, Raheem.
Thanks,
No
everyone.
problem. Thank you. Bye-bye.
Thanks.